Welcome back to PLS 510. In this lecture, we're going to start talking about nitrogen fixation. There are different ways that nitrogen from the air is fixed and made available to plants. The first I want to mention is atmospheric deposition, in that we can get a very small amount of nitrogen from the lightning source and in rain and snow, and this typically is less than about 20 pounds of N per acre per year. The way we get commercial fertilizers is through industrial fixation, or the Haber-Bosch process. This is an energy-intensive process that takes atmospheric nitrogen and makes our ammonium fertilizers. The source of energy is natural gas. And the third way, and the way that is most important to us, at least in forage crops, especially legumes, is going to be biological fixation, which is the result of a symbiotic relationship between rhizobia and legumes, where energy from the plant is used to fix the atmospheric nitrogen. And again, I want to make the point that nitrogen fixation is an energy-intensive process for the legume. So biological nitrogen fixation is carried out by legumes and specific bacteria, rhizobia, and atmospheric N is fixed or converted chemically to ammonium N, which can be used then by the plant. It's catalyzed by the enzyme nitrogenase, which contains iron and usually molybdenum, and we'll talk more about nitrogenase in just a moment. We've probably said this before in class, but nitrogen is a very important element, probably the most important element in grass systems, and it's the most limiting nutrient in those grass-based pasture and hay systems. Nitrogen is, of course, necessary for rapid growth and high yields, and it is an essential component of proteins, which forms then a good, valuable part of our animal nutrition for livestock. Nitrogen fixation as a reaction is the second most important chemical reaction in nature and in forage crops, second, of course, to photosynthesis. Biological nitrogen fixation can happen in a number of ways. It can happen in free-living nitrogen-fixing organisms like cyanobacter, but we're going to talk about the symbiotic nitrogen-fixing organisms, particularly those in legumes, for the rest of the section here. Rhizobia is the collective term for all of those microsymbionts of legumes. They are aerobic gram-negative bacteria, and the most important thing in that bullet is they are aerobic. You have to have air. It's energy intensive. They're using sugars from the plant to fix nitrogen. These are found independently within soil, but they only fix atmospheric nitrogen when in symbiosis with the legume plant. We may have been getting ahead of ourselves and using the word symbiosis without defining it, but here we will do it now. Symbiosis is a mutually beneficial relationship between two organisms. In this case, the legume is going to get nitrogen for amino acids and growth. Then the rhizobia receives energy, which comes from photosynthesis and other metabolites. And this process starts with the infection of root hairs of legumes by rhizobia, and that infection leads to the nodules that we can visibly see. And here we see those nodules. On the right are three roots well infected with rhizobia and with very prominent and numerous nodules. And on the left, you will see legume roots that are not infected at all. And if you're going to dig up plants in the field and try to diagnose how many, or try to get an estimate of how many nodules are present, you'll need to be very careful how you dig up the plants, particularly if the soil is compacted or very dry, because those nodules are onto the very fine root hairs or parts of the root system, and they're easily sheared off from those roots. A very important diagnostic feature to remember is that actively nitrogen-fixing nodules are going to be pink in color when you split them. The development of symbiosis is a three-part process, and this is how we describe it with migration, infection, and nodule development. First of all, migration. The rhizobia have to move in the soil to get to the root hair coming out of the roots of legume seedlings or some mature legumes for that matter. Those rhizobia follow a trail of root exudates. When they come into contact and the root actually will curl over, the rhizobia will congregate at the root tip. That leads to the curling of the root tip. Then the cell wall degrades and the bacteria invade the plant through an infection thread. They move to the cortex of the root and begin to branch. The nodule development is when the cortex begins to expand when infected, where it's infected, and then it produces that nodule that you can see. The bacteria grow and lose their cell wall, and at that point we call them bacteroids. The plants produce leg hemoglobin, and the bacteroids produce nitrogenase. And we'll talk about how these two enzymes work together to make nitrogen fixation possible. This is a cross-section of a nodule where you can see the majority of this picture is the nodule itself, but on the left-hand side is the root and the vascular system of the root, and you can see that the nodule has grown, infected the root, and grown quite substantially compared to the size of the root that it infected. This is going to be a little bit of a short animation of how an infection of a legume root takes place. So watch. So the root has exudates. The rhizobia are attracted to those exudates and congregates at the root tip. The root hair continues to expand and curls over, at which point the infection occurs. The infection thread moves bacteria, or the bacteria grows into the cortex of the root and congregates there, begins to swell, breaks down its cell wall, joins with the plant, and then becomes the nitrogen-fixing nodule. So let's talk about those two legumes and how they work, those two enzymes and how they work together to fix nitrogen. You've got nitrogenase, which is the enzyme provided by the rhizobia, and it catalyzes the conversion of nitrogen gas into ammonia. It is inactive in aerobic environments. Now, we have to have air, we have to have oxygen, at least in some fashion, available to those roots, because you're using a lot of energy in respiration to fix that nitrogen. And that's where leg hemoglobin comes into play. Leg hemoglobin is a heme protein, a hemoglobin-type molecule responsible for control and transport of oxygen within the nodule. So leg hemoglobin is moving nitrogen around the nodule to the tissues that need it, but at the same moment, we have got it segregated, or the oxygen segregated from the nitrogenase reaction so that nitrogen fixation can occur. A very elegant solution. There are several factors that affect how active this symbiosis reaction takes place. First of all, there's the interaction in the first place. The legumes are only infected by specific strains of rhizobia, and the plant itself determines how much photosynthate is supplied to the roots for that bacteria to use. The number of rhizobia present, that second bullet, is important. We alter that by inoculating seed when we plant it. And also if we've grown that crop in that field before, you will have inoculant present that way. The strain of rhizobia matters, both the genus species, the specific strain, but even within the right genus species, there are more and less effective strains of rhizobia. The host plant has to be actively and growing for that infection to take place. And nitrogen fertilization in very small amounts, at less than 30 pounds of N per acre per year on very, very small seedling alfalfa or clover, may actually stimulate nitrogen fixation, because it makes those very young plants that are too young to have actively fixing nitrogen nodules. It'll help them to grow actively. It'll make that infection process occur more rapidly, and then we will have more nodules sooner. If you're talking about applying nitrogen over the top to a grass legume stand, that is very inhibitory to the nitrogen fixation reaction, and we'll talk more about that in a later section, because when the nitrogen is free and available in the soil and the legume plant does not have to spend a lot of its own energy to make it, it will preferably take it from the soil. We're about to draw this part of the class to a close, or this little subsection to a close, but I do want to talk about inoculation of legume seed. While most commercial lots of seed come pre-inoculated, you may wind up needing to inoculate a lot of seed, or a group of seed, or some bag of seed, depending on the situation. So inoculation, of course, is going to ensure that we've got enough rhizobia present, not always needed, but it is inexpensive insurance. There are 4 methods. There's the sprinkle method, where we lightly moisten seeds with a sugar water solution and then add the inoculant. Now, the picture on the right is a bottle of soda that has been allowed to go flat. In other words, the carbonation is gone. So it basically is sugar water. And then next to it is a bag of inoculum. It's peat-based inoculum, a black inoculum, for both alfalfa or clover. And so in this case, you would take the soda, the sugar water, put it on the seeds, mix it around so they're lightly moistened, and add the inoculum. That's the sprinkle method. The slurry method is where the inoculum is mixed with water, or that sugar solution, and then mixed with the seed, and that gives the best distribution on the seed of the ones for this do-it-yourself methods. Now, the next one is the worst method for a do-it-yourself method, but it happens more frequently probably than we would like to think, and that's where dry inoculant is simply added to the seed box once the seed's in it, and you mix it around and you seed. And as I said at the beginning of this slide, the most common way that we get inoculum on seed these days in commercial seed lots, especially of improved varieties of legumes, is by a coating on the seed. So we call that pre-inoculation. And the inoculant is already applied, is already on the seed in a lime coating, and that's the most common. Also in that lime coating are going to be quite frequently fungicides, which will prevent some of the diseases that will attack seedlings while the plant is trying to get established. Last point I want to make is that there are specific cross-inoculation groups depending on the legume. For the alfalfa group, that's alfalfa and sweet clover, the cross-inoculation group, or the right kind of rhizobia, if you want to think of it that way, is Cynorhizobium melloloti. For the clover group, it's Rhizobium leguminosarum. There are the other ones listed down below, but primarily we're going to be talking about these, or you're going to be using these in the field where you're to be planting a legume. And as that previous slide showed a couple back, you may find them mixed in the same bag of inoculum. So I'm going to draw this subsection to a close and let you know that next is going to be, we will have a discussion of the movement of nitrogen in grass legume systems. So we'll see you then.